Assalamu alaikum, everyone, and Ramadan Kareem. I am Farah Abul Rub. I'm a third year medical student, and for today, we're going to be going over the hormonal regulation of spermatogenesis as well as some sperm defects. For this session, I will be going over a basic overview of the functional anatomy very, very briefly, just to basically connect everything together. And then we will be going over to the actual bulk of the session, which is the spermatogenesis or the spermatogenesis, however you'd like to say it. Then we'll be moving on to the male infertility as well as the actual abnormalities in sperms. And finally, we're going to have a few practice questions just to test your knowledge and make sure that you guys are on the same page. And if you have any questions for me at any point, feel free to stop me and let me know. Okay? Now, for our functional overview, since you guys will be starting the female reproductive system very soon, I am trying my best to kind of pull you back from the big picture of just fertilization and what happens at the end result where we meet the actual ovum of the female, okay? So we're going to take this in reverse, and then hopefully by the end of the session, you'll be able to understand why everything happens the way that it does, since we're already familiar with the entire fertilization process, the entire weeks of embryology, so on and so forth. So I thought that would be a good place for us to start. Now, where does sperm actually come from? Sperm is basically something that is produced in this sac that you guys see right here, which is the testes, okay? It is in the scrotum or the scrotal sac. As it gets produced and as it's getting developed, let's say, it is going to pass through multiple structures in order to eventually be ejaculated. So this is a nice mnemonic that I personally think helped me. And you, as disgusting as it may be, you can think of 7-Up. So seminiferous tubules, and then it goes to the epididymis, and then it goes to the vas deferens, which are those tubes here that we can see. Then it is going to reach the seminal vesicle, obviously, but that's not in the mnemonic, so just don't worry about it. And then the ejaculatory ducts, which open into the prostatic urethra, right? You guys did renal. And then none is just for the mnemonic. And then the urethra itself, actually, and the penis, eventually, where it will be ejaculated. So let's take a closer look at the site of production and the factory that we have in the actual testicle. Here we can see a cross-section of the testicle where we have those kind of lobulated structures surrounded by a meshwork, okay? Within this meshwork, we are going to find what are called Leydig cells that primarily respond to LH, and we're going to have those peritubular cells. Now, this type of orientation is very important because sperm is basically viewed by the body as something that is foreign. Now, let's keep in mind that the egg itself, the sperm, and even the fetus are all basically considered parasites to the immune system and to the body. So we need to have a very clear blood testes barrier or a blood sperm barrier in order for us not to have any sort of mixing that would contribute to an immune reaction, to breaking down of those sperm, or to any sort of abnormality, let's say. Now, within those lobulated structures that you guys can see here, we have what are called Sertoli cells. Sertoli cells are more responsive to FSH and testosterone. Sertoli cells are basically cells that nurse the sperm that are around them. They have three main functions that are supportive, exocrine, and endocrine. Their supportive functions are mainly for maintaining and reforming tight junctions to make sure that the sperm aren't getting in contact with the blood, thus they are maintaining the blood-testis barrier. They're also involved with phagocytosis of any of the cells that are dying, and thus they are producing nutrients by breaking down those cells for the cells that actually need it, okay? Now, for the exocrine functions, think of this as the actual very male type of function, right? So what is the actual purpose of those sperms? We want to ejaculate them, right? We want to have testosterone. We want to basically produce the seminal fluid. And that is exactly what the exocrine function is. So it's going to produce the fluid to move the sperm that are immobile until they do become mobile, which is basically the seminal fluid. And it's going to produce androgen binding proteins that are for testosterone. Lastly, it is going to basically determine the release of the actual sperm once it is time for ejaculation. The endocrine functions are the somewhat more passive functions of Sertoli cells, where they are going to be the ones that are expressing receptors for the follicle stimulating hormone. They're going to be expressing things that will suppress the female counterpart of this entire situation. So the Müllerian inhibiting substance or the anti-Müllerian hormone. They're also going to have the actual transfer of testosterone to estradiol. So this is why it's somewhat passive. We have things that are not directly related to just the male here, right? We're talking about the anti-Müllerian hormone, we're talking about the estradiol. So those functions you can keep in your mind. Okay, so it's not necessarily all just male, male, male. This is going to be endocrine, whereas exocrine is just purely very male type of functions. Is this clear so far? Okay, I don't see any questions, so hopefully it is clear. Let's move on. Now, the actual sperm itself that we have is composed of various different structures. It's a tiny little thing, but it has so many details to it, okay? So the head that we can see here includes the nucleus of the sperm and the genetic information. Moving down, we can see this mid piece, okay? That is going to include the spiral mitochondria that basically provides the energy for this flagellum that we see here. So this mitochondria here is in the spiral type of formation in order for it to consistently generate ATP, which will propel the sperm forward in order for it to actually be able to reach the ovum. Now, the ovum, once you guys take it in more detail, you're going to realize that it has so many protective coatings around it. So we need to be able to overcome those. And the way that we do that is with this layer here called the acrosome. The acrosome has the generative enzymes, including hyaluronidase and acrosin, which is a protease, and they break down this layer of cells that are on the outer membrane of the ovum called the zona pellucida. Okay? So through all of these structures that we have here, we realize that, okay, so the acrosome is basically going to penetrate into the ovum, right? And then it's very conveniently placed such that the nuclear material and the genetic material can be just propelled into the ovum immediately after. And then the mid piece is basically our engine, and our flagellum is the wheels of this operation. So as this mitochondria is producing ATP consistently, it will propel this sperm so that it is able to actually move. But what actually makes the sperm move? Is it just like something from Allah subhanahu wa ta'ala where the sperm knows to go to the ovum? Well, kind of. But to put that into more scientific terms, we have a thermal type of gradient that attracts the sperm, as well as a chemical type of gradient that also attracts the sperm. Okay? So this thermal gradient is going to be called a thermotaxis. Think of an actual taxi that is driving and it's taking you around, or a chemotaxis, which is related to the actual chemical makeup within the vagina, within the uterus, and the fallopian tubes. So the process upon which those sperm can actually reach the ovum is called chemotaxis, and it creates this gradient so that the sperm can be attracted to the ovum and so that they can actually reach it. Now, in order for this chemotaxis to take place and in order for the sperm to become motile, they need to undergo a process called capacitation. What is capacitation? Capacitation is basically modification of the sperm such that it is able to actually fertilize the egg. So when sperm is ejaculated and before it reaches the vaginal canal, it is going to look like this. A lot of what we need, a lot of the good stuff is covered up. So within the vaginal canal, due to the pH, due to various different enzymes, you're going to have this capacitation reaction where it's no longer decapacitated, it is now capacitated. And we're going to have the acrosome actually exposed. Now, what type of modifications occur in order for us to move from this decapacitation to the capacitation? I like to remember it using this CCGP. And remember that the C and G is for basically less, but the ones on the outer are for more. So I am increasing permeability to calcium. I am decreasing cholesterol such that the permeability increases as well. I am decreasing glycoproteins and I'm increasing phosphorylation. Okay? So through this process, I am able to basically uncap the sperm, and now it is the process of capacitation, and we have the sperm that is ready to reach our ovum. Okay, we said that now the acrosome is exposed, we're ready to go, but what does the acrosome actually do? It participates in this acrosomal reaction where it releases enzymes, it perforates into the zona pellucida, which you guys can see here, okay? It wants to perforate into the zona pellucida, and in order to do that, it is going to secrete its enzymes, its proteases. So without this acrosome, really, the sperm cannot do much, right? It does not have those digestive enzymes. It does not have those proteases. And so it would just basically, it's a blunt type of surface trying to shove against something else. It's not going to do that much damage. It's not really going to be able to penetrate much, which is why looking into the future now, when I'm talking about sperm defects, if something is missing an acrosome or if something is not the correct head shape, I'm going to start to think, okay, so it doesn't really make that much sense for it to actually be able to fertilize, right? And that brings us to the end of the first section. Moving on to the actual spermatogenesis or spermatogenesis, I wanted to, as I mentioned, just take you guys over the very big picture that you guys already know. And now we're going to look at the backstory of our sperm that was able to reach the ovum and successfully fertilize it. The way that sperm is generated and the way that sperm is created is through a process called spermatogenesis. It is the process of germ cell differentiation to produce spermatozoa. It begins at puberty and it is protected by the blood testes barrier. Okay? It typically takes an average of about 72 days and it is regulated by FSH and testosterone. Why? Because it is regulated by the Sertoli cells, right? They're the ones that are actually responding. Spermatogenesis can be broadly classified into three distinctive states or three distinctive steps. First of all, you're going to have division, which is also called spermatidogenesis because we're creating a spermatid. And then after spermatidogenesis, I want to mature the spermatid that I had. So I'm going to have spermiogenesis, okay? Lastly, I want to actually release the sperm that I had, and this is in the process of spermiation. So first of all, I'm creating a spermatid in spermatidogenesis. Then I'm actually creating a sperm, a fully mature sperm in spermiogenesis. And lastly, I release it using spermiation. So let's take a very quick overview of what the actual cell division process is, because we will be using both of those processes, including mitosis and meiosis. I'm sure you guys remember this better than I do, and you know this very, very well, so I'm not going to spend a long time on it. But mitosis is basically the process of all somatic cells. Every single cell in the body undergoes mitosis, except very, very few. But almost all cells in the body are going to undergo mitosis, where our now replicated 4N cell is going to split to give me two 2N type of cells that are absolutely identical to the mother cell. Okay, so we start off with 2N, we replicate 4N, and then we split into 2N again. So we have a diploid cell at the end that is identical to the mother. Meiosis is the process that is undertaken by the germinal type of cells. So this is everything that we can see in sperm and in ovums, and it's only in those types of cells. Meiosis is the process wherein we start off just like regular mitosis, okay? However, we have an extra step, which is meiosis 2, where we take this 2N cell and we split it again without actually replicating it. So we don't do a duplication where we move this once again to 4N. We just immediately take this 2N and split it into 2, okay? So here we had a 4N that split into 2N and 2N. Here we will also have a 4N, but it goes into 2N and then into just N. So we're going to end up with a haploid type of cell. Now, these cells are actually genetically different and genetically unique compared to the mother cell. And that is because of a lot of processes that happen. You guys are also taking the genetics parallel, so I'm sure you know the details, where you have this cross matching, where you basically take a chromosome, a portion of it, and you give it to the other one, so on and so forth. So you're creating unique haploid daughter cells that are different from the mother cell. And you end up with 4 cells instead of just 2. Okay? Now, since this is a somewhat delicate and complicated process, it can often go wrong. And this does happen because the process that we have, where we're trying to split the chromosomes evenly, might go wrong. We can have a process called non-disjunction. Okay? So disjunction is the typical process that we usually have. It is physiological, it is normal, it is what we should have. Non-disjunction is when something goes wrong. So we could have non-disjunction in meiosis 1, where we end up having all the chromosomes in 1. So this is going to be 4N, and this is going to be nothing. And ultimately, you're going to have 2N cells. And here you're also going to have nothing. On the other hand, we can have non-disjunction in meiosis 2, where we do have normal meiosis 1, right? 2N cells. But when they split, something happens where instead of me getting 2 haploid cells like normally here, I'm going to have 1 diploid cell and 1 cell with nothing. So this cell that doesn't have anything typically is incapable of actually fertilizing because it doesn't have any genomic material. However, these cells that are 2N can actually cause fertilization. And this is what happens to us in Down syndrome and Klinefelter's. Down syndrome is when we have trisomy, or 3 chromosomes instead of 2, so a set of 3, of chromosome 21. Klinefelter's is when we have an extra X chromosome. So instead of having 46 chromosomes, we're going to have 47, and it's going to be XXY. So we have an extra X chromosome. Is that clear for everyone so far? Any questions? All right, I think we're good. Okay. Now, to get to the actual process of spermatogenesis itself, which is this umbrella term that we categorize into 3 different stages, we have this kick-starting in embryonal life. So since fetus, much English, since a person is in their mom's belly, since in utero, you're going to have this germinal cell line proliferation. However, you will not begin to undergo meiosis until puberty, okay? Talking about purely the male here. So in the embryo, you're going to consistently try to create as many germinal cells as possible. So we start off from a stem cell, a germinal stem cell called a spermatogonium, okay? Or spermatogonia as plural. So this cell is going to consistently replicate and multiply using mitosis to give me multiple spermatogonia, okay? These spermatogonia are identical to the mother cell and they are diploid, so 2N. In puberty or in reproductive years, these spermatogonia are going to actually be thrusted into the spermatogenesis, where meiosis begins. Our first step is to make it a 4N type of cell, right? That's how we start both meiosis and mitosis. But now that we're actually kickstarting the spermatogenesis, this is going to be called a primary spermatocyte. This primary spermatocyte, after meiosis 1, now that we have a 2N type of cell, is going to give me a secondary spermatocyte. After finishing meiosis 2, the secondary spermatocyte is going to give me haploid spermatids, which will move on to the next process of maturation where we're creating our sperm, right? So spermatids mature into sperm. Both of them are haploid. It is just a process of maturation, which is in spermiogenesis. Lastly, as you guys can see, we're basically progressing downwards, and this is going to be our lumen of that lobule that we looked at. Over here is when we're going to have our spermiation, which is when we actually release this sperm into the lumen such that it is able to move from the seminiferous tubules into the epididymis, so on and so forth, our entire process. Let's take a look at that on a bit of a closer scale. So here we have our basal cell layer, where we're going to have our germinal type of cells, our stem cells, which is our spermatogonium. As we progress more towards the lumen, we can see that we're more advanced. We are more progressive in our actual sperm stage, and we can actually release a fully developed spermatozoa into the lumen, okay? So spermatogenesis is the process whereby I take my spermatogonium and I make it reach a spermatid, okay? So spermatidogenesis, that's my end product. I want a spermatid. I start off with my spermatogonium or my spermatogonia, okay? So because multiple of them are doing this process at the same time. It becomes a 4N cell following DNA replication to give me a primary spermatocyte. This primary spermatocyte will undergo meiosis 1, which is similar to mitosis, and give me a secondary spermatocyte, which is diploid. And then after that, this secondary spermatocyte will undergo meiosis 2 to give me my spermatids. So the process from the spermatogonium to the primary spermatocyte is basically mitosis, right? We haven't actually started meiosis yet. However, once we transition from the primary spermatocyte to the secondary spermatocyte, we are actually doing meiosis 1. Eventually, we're going to have our spermatids, which are our haploid, and they are what actually gives us the end product that we want. Now, this goes on in cycles, and this continuously happens to multiple cells all at once. And this overlap basically ensures that we have a residual population of the spermatogonia that maintain the ability of the testes to continuously produce sperm. Okay, we have our spermatids. What is our next step? Our spermiogenesis. I want to move the spermatid from this very primitive type of structure into an actual, fully developed and motile sperm, right? So spermiogenesis. The process of spermiogenesis is basically when I'm trying to change the morphology of my spermatid so that it resembles more closely my final and end product of a mature spermatozoon, as you guys can see here. How does it achieve that? First of all, as you can see here, the nucleus is pretty large, right? And my nuclear material is going to be very dispersed all over the place. So it's going to condense in order for it to also be more easily expelled into the ovum after. And it's going to, as you see here, take up this type of shape. Why is it taking up this type of shape? What are we forming? The acrosome. So I formed this acrosome, but as you guys can see, it is still covered because it is decapacitated. I formed this acrosome that is going to participate in the acrosomal reaction later on. Now, I'm at this stage, right? I formed the acrosome, I condensed the nucleus, but I'm still not a motile type of sperm, right? So I will start to reposition the sperm and move it around such that I'm able to create this elongation that is going to be the flagellum or the tail. Okay? When we have the flagellum, we're ready to go. However, we need a lot of energy, right? So far, all that we really have is just DNA or nuclear material and a tail. Where am I going to get the energy from? We get the energy from the mitochondria, which creates this spiral type of structure here. And as we said, it's going to produce ATP, which will create this propelling type of motion for the sperm. Okay. Now, lastly, the sperm is going to realize, okay, this is a survival of the fittest type of situation. I need to get to the ovum. I need to be very quick on my feet. I need to be as light as possible. So in order for it to maximize efficiency, it is going to get rid of any external excessive cytoplasm that it no longer needs. This sperm is a very, very, very efficient type of cell, right? It just has all that it needs, really, a nucleus with the nuclear material, a tail that can propel it, and energy for that tail. It doesn't really need much else. So it gets rid of this excess cytoplasm as a residual body that you guys can see here, okay? Through this process, we have now developed from a regular spermatid that was also haploid into a sperm, a fully mature spermatozoa or a sperm. Okay? Is that clear so far? Any questions? All right, we're good, inshallah. All right, our third and last step in our spermatogenesis is spermiation. So spermiation is this propelling into the lumen of the lobule that I showed you guys. Okay, where we break down the Sertoli cells, sperm cell junctions that the Sertoli cells were creating to nurture those cells and to protect them. And we basically release those sperms into the lumen so that they can be then ejected into the epididymis and move on to eventually be ejaculated. Okay, so very, very straightforward. We have three processes, as I told you guys: spermatidogenesis, Spermiogenesis, and finally spermiation, where we initially start from a spermatogonium into a spermatid, and then we move from a spermatid into an actual sperm, and lastly, we propel this sperm outside and into the lumen of this lobule that we have. And that is basically it. We are done with spermatogenesis. Now, moving on to our last portion, which is male infertility. What is infertility in general? Infertility refers to when you have had a failure to conceive after 12 to 24 months or more, so a year or two, of regular, unprotected intercourse. Okay, so the couple is actively trying, but it's not working. Typically, people might think, okay, so infertility problems are usually more related to the female, and she should be the one doing all the tests and all that, but that is not the case. More often than not, infertility is actually unexplained or idiopathic, as you guys can see here. Sperm problems and ovulatory failure both do have very, very large contributions as well. Now, with regards to male issues specifically, as you can see, idiopathic causes and testicular insufficiency are equal. So if you were to get asked what is the most common cause of male-related infertility, it would either be idiopathic or unexplained or testicular types of insufficiency. Now, for the sake of this session, I broadly categorize the types of the causes, let's say, of infertility that were mentioned in your lecture into gross structural abnormalities and spermatic structural abnormalities. For the gross structural abnormalities, the one that you guys have to really focus on and know is the varicocele or the varicose veins. These varicose veins are basically engorged veins that are surrounding the epididymis and the testes. This could happen to a variety of different causes. I'm not sure if you guys are going to go into the details of them, so I don't want to overwhelm you with those. But let's just say we have some kind of obstruction that is stopping the flow of the blood back. This would lead to this type of engorgement and somewhat of a bag of worms type of appearance on palpation or even just inspecting the general area. Okay. Now, this type of gross structural abnormality is making the person more prone to develop sperm that have coiled tails. So the way that I like to think about it is these veins are super, super engorged, right? They're all over the place. They're coiling around each other. So the sperm, in order to try to move out from the epididymis and the vas deferens, is going to also try to coil around it. So it's going to have a coiled tail type of appearance. Other things that might contribute to growth structural abnormalities are temperature as well as testicular cancer. Now, cryptorchidism was not mentioned in this specific lecture, but I'm sure you guys will cover it. So, cryptorchidism is basically when the testes do not descend properly, either one or both of them. And they can stop at so many different places. So it could be all the way up in the abdomen, it could be in the inguinal area, it could be basically anywhere along the route of descent. Now, why cryptorchidism is very important is that the reason why the testes are actually in the scrotal sac outside of the body is that they are very, very sensitive to temperature, and sperm can very easily die at very high temperatures. The abdomen and the abdominal cavity, if those testes were stuck in there, would actually cause those sperm to die. So the same applies for when the body temperature increases for any type of reason, including fever, for example. If someone has a fever for a prolonged amount of time or a very high grade type of fever, so 40 degrees Celsius, let's say, they might be susceptible or they are at risk of developing this type of defect, okay? Now, testicular cancer is also commonly associated with abnormalities that are reflected in the actual sperm as well. So you might have microcephalic spermatozoa, which basically means that the head of the sperm is way too small. You might have acrosomal defects. You might have excessive residual cytoplasm, which basically means that we did not have that residual body that was created, right? We could have very short tails, which would hinder this type of sperm's motility, right? All right. Now, moving on to the actual spermatic structural abnormalities, we have so many different types, but the one that you guys should focus on is the globozoospermia. From the name, it's basically a globe type of structure or a balloon on a string type of structure where it forms this very rounded head that doesn't have this pointed edge that we see normally, okay? Other types of structural or morphological abnormalities that you can see is this microcephalic head, this very, very small head, a condensed acrosome, which might still be able to fertilize, it's just a bit more difficult, a double-headed type of sperm that did not separate properly, a double-tailed type of sperm, a very large-headed type of sperm. So all of these are very, very commonly seen. And actually, since we are producing hundreds of millions of sperms, we will actually be able to see so many of those, and it might not necessarily mean that the patient is going to have infertility. Due to the huge number of those sperms, they can still be able to conceive despite those morphological abnormalities if they have a large enough number that are actually normal, motile, and sufficient. And that actually brings us to the end before we move on to practice questions. So, do you guys have any questions at all? I told you it's going to be a super short session, inshallah. It's very, very quick. I really hope that it was clear and you guys understood. Do you have any questions for me at all? If you don't have questions, just say no or clear or something so I know you guys are with me, please. Because I'd like you to answer for the practice questions. They're not that many. All right, no response, but it's okay. We'll move on to our last section, which is the practice questions. So I tried my best to include something about almost every point that we've mentioned. Our first question is a 54-year-old male that is admitted to the hospital with severe back pain. Upon radiographic examination, which you guys can see here, the scrotum resembles a bag of worms. Which of the following conditions will be most likely associated with this radiographic picture? Varicocele, rectocele, cystocele, hydrocele, and hypospadias. What do you guys think? I don't know if you guys are thinking or if you're just not gonna answer. So, okay. So a bag of worms is a very, very good clue that you guys might see in questions that basically tells you that, okay, this is going to be varicocele. And as we can see here, these veins are very tortuous, they're very engorged, they're not normal whatsoever, and they are surrounding the testes. So this is going to be varicocele. Rectocele is for the rectum, cystocele is for the bladder, and like you don't need to worry about those, but just in case you get the names mixed up a bit. So varicocele is the one that we're talking about. It is varicose veins. Our next question is to identify the following sperm defect. A is globozoospermia, B is microcephalic heads, C is asthenozoospermia, D is residual cytoplasmic material. All right. So as you guys can see here from the head of the sperm, it kind of looks like a globe, right? It's very, very rounded. It's very smooth. Doesn't have any type of head, this pointed head that we typically see. So this is the globozoospermia. Microcephalic head means a super tiny head, okay? So this isn't the case here. Question 3, which of the following cells undergo meiotic division? A, primordial germ cells, B, primary spermatocytes, C, secondary spermatocytes, and D, spermatids. So it's meiotic division, not mitotic. All right. So the answer for this one would be B, the primary spermatocytes. They are the ones that are going to kickstart this meiotic division process where they will have this 4N and then through meiosis 1 give me the secondary spermatocytes, then through meiosis 2 will give me the spermatids. Okay, so although the secondary spermatocytes and spermatids are involved in the meiotic division process, they are not the starting cells, they are the end products, let's say. So spermatids will not undergo any type of division further. They are already haploid. Our next question is the formation of mature sperm cells from a primitive germ cell takes an average of, this is pure recall, this is C, 72 days approximately, 72 to 74 days. Next question, a hormone secreted from Sertoli cells will act directly on which of the following? A is LH, B FSH, C inhibin, and D estrogen. For this question, it is going to be B FSH. Inhibin is secreted by the Sertoli cells as an endocrine type of function, right? But the primary things that a Sertoli cell acts on is FSH and testosterone, not estrogen. All right. Our last question is spermiogenesis results in the metamorphosis of which cell type? A, secondary spermatocyte, B, spermatogonia, C, primary spermatocyte, D, spermatozoa or zoan, and E, spermatid. So spermiogenesis is going to be the conversion or the update, basically the upgrade of a spermatid into a spermatozoa or a full-on sperm. So the answer would be E. And that is that. We are officially done. For the one person that is in the session, I'm not sure if you're actually in the session or not, but feel free to please scan this QR code to give us your feedback. I'd really appreciate it. I really, really hope that this session was beneficial and it was very straight to the point. Best of luck with your studies, inshallah, and enjoy the vacation if you are able to. I'll be here for any questions for the next few minutes. Thank you. Okay, so you are actually here. Was the session beneficial, inshallah? Was it clear? Okay, I'm glad, alhamdulillah. Please scan the QR code if you're able to give your feedback. And I'll be sharing the slides with you guys as well if you need them.